Okay, can everybody see the presentation there? You watching it on Teams? We have a look in the chat. See if... Yeah, working?
Yeah, it's working
Great.
now. Thank you.
Fantastic. Okay, well, it's a real pleasure to be here this morning with some people in front of me. So I have some real people in the audience, which is a real treat. And I think the first time I've actually stood up and given a talk in the last 2 years. So it's a real pleasure. And thank you everybody for coming here in person. And welcome to those of you who are unable to come and are joining online. It's great to see you too. So I'm going to talk this morning about some work we've been doing on detecting soil degradation and restoration, and it's largely based around a project that myself and Jason Neff from the University of Colorado put together a couple of years ago, and we're very actively working on it. So what I'll show you this morning is very active research, not the kind of final polished product. But I think it links very nicely to the theme of the meeting here today. So we have a project which is funded by the British Research Council, the Natural Environment Research Council, the National Science Foundation in the US, and the USDA. And it's funded under the Signals in the Soil program, and it involves scientists from Lancaster University, Manchester University, and the University of Colorado. And there's quite a big team of people working on this project. And one of the really interesting and exciting things about the work we've been doing is that we've involved people who not only work on soil science, and we've got a great group of us, of soil scientists in the team, but we've also included people who are experts on sensors and data networking and on data science. So we've brought together this very multidisciplinary group of engineers, material scientists, mathematicians and statisticians. And I think that combination has made it a very exciting project to work on. So what I'm going to do in the talk this morning, I'm going to talk very little bit about soil health, since that's been the kind of the driver for our sensing work. I'll then talk about some of the work we've been doing in the project. So the next steps in detecting and predicting soil change. We'll look at some new sensor development around soil biology sensing. We'll talk a bit about how we've been trying to extract information about soil functioning from soil data. And then I'll finish up talking about how we've been trying to look at scaling up modeling, monitoring and predictive capacity, looking at integrated sensor development and satellite communications. And then we'll kind of wrap up by thinking a little bit about the future. So many of you working in the field will know that there are a myriad of definitions of soil health out there. And I've just put a few of them on the slide here, and I'm not going to go through them. You'll be glad to know. But I think that it's one of those kind of phrases which has caught the public's attention, and perhaps the scientific community has struggled a little bit with this whole context. But I think if we think about it, and I've just outlined the one at the top there, which is the definition which tends to, I think, most people tend to adopt, is around this continued capacity of the soil to function as a vital living ecosystem that sustains plants, animals, and humans. And so it's that biological component that we see as being very important, this living soil concept. And so this importance of soil biology has really kind of taken off in the scientific literature. There's been a number of reviews published recently, and I've just put a few of them on there, that attract a lot of attention. If you look at the total views on some of these papers, they're really picking up attention. And I think not only from the scientific community, but also from the general audience. So soil biology is important. The other thing that I think is really important and has really come to the fore is this multifunctional nature of soil health. So, you know, when I did my degree in soil science many years ago, it was all about production agriculture, about learning how to manage soils to produce more food. And I think what we've seen over the last 20 to 10 years is this recognition that soils are multifunctional, and that if we're thinking about soil health, then we also need to think about this multifunctional nature of the soil. And there's been some interesting papers published over the last few years. This one from European Journal of Soil Science, trying to, where they went out, I think, and took about 90 odd different soils and looked at their contribution to different soil functions. And you can see the functions there along the bottom of the slide. Get a pointer here. Primary productivity, obviously important, water regulation, biodiversity, nutrient cycling, and climate regulation. And then they classified the contributions of these soils made to those different elements. And you can see differences here between grassland and arable soils, grasslands having many of the soils contributing highly to climate regulation, less so for arable soils, but arable soils contributing highly to water regulation. So you can see these differences being pulled out between these different groups of soils. And this has resulted in the development of a large number of indicators. So as we've tried to say something about soil health, how healthy is this soil, we've gone out and done what all good soil scientists will do, and that's measure stuff. And so we've tried to look for different indicators of biology, organic matter, chemistry, and physics. And like most things in soil science, this involves us going out, taking a sample, removing it from the soil, going back to the lab and measuring something. And these indicators were taken from a recent review in Nature Reviews. What you find is if you look across all the indicators listed, there are only 4 that I could find that are measured in situ in the field where the soil is interacting with the rest of the environment. So earthworm numbers, greenhouse gas emissions, penetration resistance, and infiltration. And you could argue even with some of those that you're actually disturbing the thing that you're trying to measure. I guess this was the kind of the basis for our ideas around this project was that we wanted to get away from removing soil from the environment in which soil sat, to be able to look at the soil and see how it changed through time. Clearly you can detect change here, but you need to keep going out to the same area, and you need to keep taking samples, and you need to account for the spatial variability and all the other confounding factors. So the dynamics can only be considered by repeat sampling. So what's the next step? Well, we've been playing around with sensors, and it's great we've got Delta T here today, because I think the first water sensors I ever used in the field in Spain in about 1990 something or other was a Delta T probe. And we've been using this kind of these kinds of sensors for a long time, and we've also had the capacity, certainly over the last 10 or 15 years, to pull that information through modems, through the telephone networks, and display it on websites such as this one. This is from one of Delta T's competitors, but Campbell Scientific. But it's this, you know, most sensor manufacturers have this capability now. We're also seeing national and international groups using sensors in the field and then providing that data online. And a good example of that is the NEON data set from the US, where they've invested huge, eye-watering amounts of money. I think the budget is like $30 million a year to run a whole series of sensor-driven sites across the US. But that data is all fed up onto a web server, and you can pull it off and play around with it. And it's great to see people in the audience here who have a lot of experience in doing that. And then the other thing that's occurring is this miniaturization and the reduction in cost of sensors. And, you know, we're seeing, you know, you can go on Amazon and you can buy yourself a little kit to build an Arduino weather station for around $30. So these developments, you know, got us thinking about, could we use these high frequency signals from sensors to tell us something about the current state and trajectory of soil health? And could we think about how we could use some of these cheaper approaches to provide sensing platforms which were more accessible to a wider number of people? So I'm going to talk about the project, which has three interconnected components. I'm going to start off talking about novel sensors and these multifunctional sensing platforms. So this is about getting hold of data. And then I'm going to talk a bit about how we then use that data and the kind of work we've been doing with our data scientists to try and understand that data and what it might mean. And then talk a little bit about how we might go to scale and the kind of satellite communications that we have access to. Let's think about some new sensor development. And the two areas we've been very active in the project is around looking for something which would tell us something about biological activity, and also something which would tell us something about carbon accumulation. Took our inspiration for the microbial sensors from the T-bag index. So some of you will be familiar with this, some of you won't, but it's basically a method that was developed in Holland, where you take a couple of tea bags. One of them is a red bush tea bag, and one of them is a green leaf tea bag, and you bury them in the soil. And then you come back 3 months later, you dig up your tea bags and you weigh them and you see what the weight change has been, so how much decomposition you've had. And by doing that, you can derive a couple of parameters which will tell you something about the rate of decomposition in that soil. So our thought was, could we do something similar? And here we've developed some sensors which are based around the principle of a fuse. So you have your fuse in all the plugs in the building. We'll have a little fuse that blows when there's too much current through it, and there's then obviously a switch, and if you were sensing that circuit, you would know that circuit had broken. And so the idea was to use that approach and bury a little fuse in something which was biodegradable. So we looked at three different traces. I don't know if it's not going to work. Okay. So you won't see the fuse blowing in the little video, but never mind. We looked at three approaches. One was a conductive trace, which is basically to bury the conductive material in a material which is biodegradable. We looked at encapsulating the trace in a biodegradable material. And then we looked at having a substrate which could be attacked and was biodegradable could be decomposed by the microbes. So there are three different approaches. The conductive trace involved some carbon particles which were bonded in this unpronounceable chemical, and what you see is that over time the resistance changes, and as the trace is completely broken, it shoots up, and you can monitor that. Approach 2 involved coating these traces in a beeswax, soil wax blend, and what we found was that when you buried these traces, and you can see the kind of prototype traces there in soil with a thickness of 0.5 millimeters of this wax coating, at some point the resistance again goes up through the roof, and as the trace is broken. Where it's submerged in a buffer or in sand, you can see that the resistance is only just gradually changing. And it made quite a lot of difference as to what mix of soil wax and beeswax we use. So the percent of soil wax seemed to have a big influence on the time to failure. So there's some work thinking about exactly what you coat these sensors with. So we were very excited by those results. So we decided to deploy some at the University of Manchester. So these are kind of the prototype 2 of these little biological sensors, and you can see here the trace buried behind the wax, and then our colleagues in Colorado then link these up to a little transmitter so that all this information, through the wonders of cell phones and Wi-Fi, is linked up and then viewable on a web server. They made these ones with a slightly different mix, and they also made them 1 millimeter thick. And unfortunately, we've had 8 sensors in pots of soil from Colt Park, which is a site in the Yorkshire Dales in England, and we've had no change in resistance for 3 months. So none of them have gone yet. So not quite what we were hoping for. So a bit more learning there. Approach 3 is the biodegradable substrate approach, and you can see here the impact of time on the substrate, and the biology eating through it. And again, the idea is that through time the resistance would change, and so we've worked with these, and the great thing about these ones is they're really fast to produce and really cheap to produce. You basically just print them on a sheet of this material, but we've had some issues with basically the material being degraded, but the trace not being degraded. And so there's some work there to think about what we do in terms of making these fail consistently. And one of the thoughts we've had is to put them under tension, perhaps to have a little spring that's pulling them apart, and to get them to fail. So we're not there yet. We have had success with all of them, but we've also faced challenges with all of them. So the conductive trace works very well when it's in solution. It doesn't work particularly well when it's in soils. The encapsulant seems to be very promising, but the wet wax blend and the thickness of the wax is absolutely critical. And then the substrate one seems very fast, very promising, but again, we're not getting consistent failure, and we need to work on how to produce that. Those are the kind of activity sensors, and then I just wanted to share with you some really kind of new results that we've been working on thinking about carbon accumulation in soils. And here our colleagues in Colorado have been using some novel hydrogel composite materials. So these are basically materials with high water permeability that bind clean clay. So you're basically binding clay into this material, and it can absorb water and carbon. You can see some traces here, and you can see this project's obsessed with tea. They've submerged these sensors in tea and looked at the changes in the properties of these material over time, and the detection there is looked by measuring both capacitance and the optical properties of this hydrogel. So there's early days, but some interesting results there. Okay, so we've got some nice, interesting results from some novel sensors, but we also have been working on trying to extract information about soil functioning from soil data streams. And here we've worked with our maths and stats department here at Lancaster, looking at two things. So one is change point detection, and then initially we've been looking at dry down modeling. So we've been working here with soil moisture data. We've used data from the NEON project in the States. And what we've been trying to do is to try and derive properties that might tell us something about the soil from this data. We've used an approach called change point detection and developed new methods of working from that. And the idea there is that you run these methods and they pick up the peaks of the soil moisture and then model the drawdown curve. So this is the drying curve. And the idea is that we can then derive some characteristics of that dry down curve. So we can calculate something which describes the rise in soil moisture at the change point. We can derive something which tells us how fast it decays, and we can also extract the asymptotic soil moisture content. So the idea is that this is done automatically, so that the data is read in, and then the change points are identified. So this is some real data, and you can see all the change points there with the little triangles, and then we can derive these.
Našel nějaký asistu, nevím, jestli to zaregistroval jako od Zuzky Peštový.
Sorry, but you're muted, I guess, or at least I don't hear anything anymore.
Yes, you're sound like you muted yourself, John. Sometimes it happens to me when I'm changing the slides. Yeah, now you're unmuted.
That is weird, isn't it? Okay, thanks very much, folks. Sorry about that. I will continue. No. Can everybody see the screen okay and hear me? Yeah, good.
Yes.
Okay, so we can develop this kind of dashboard. The final part of the project is the kind of scaling up and the predictive capacity. So the final element has been around developing some field-ready multi-sensors and then the satellite communication work. So our field-ready multi-sensors are trying to utilize low-cost technology. So we've been working on weatherizing CO2 sensors. So these are little commercial sensors which are inexpensive compared to the ones we have in the field at the moment, which are around, I don't know how much they are, but they're frighteningly expensive. Probably about €1,000 a pop, whereas these ones are probably about 30, 40 euros a pop. So we're looking at producing these combinations of sensors. So we would have a CO2 sensor, a soil moisture sensor, and a temperature sensor, and we hope that we'll be ready to trial these in the field in the next few months, and hopefully by the summer. And then the final element is around how we communicate with these sensors. And so we've been working with low-cost satellite company, Swarm, which have launched these little micro satellites. You can hold them in the palm of your hand, and you can send small amounts of data to these satellites at a very low cost. And so our colleagues have already been doing this around monitoring water wells, in checking whether a water well is working, so that maintenance teams can be sent to remote locations. And so we have the radio developer in our team, and we're ready to use this with these sensors, and we just need to now get the sensors into the field so that we can use the satellites. So to summarize, we have these soil biology sensors under development. I think we're making some really exciting progress there. We've made some initial steps towards carbon accumulation sensor, obviously with potential application in soil carbon monitoring. We have been working on the data analytics for soil water, and we have some of those developed. And the focus is now shifting towards CO2 to see what we might be able to do there using these data science approaches. And we hope to have this low-cost multi-parameter sensing technology by the summer, and access to this low-cost satellite network. We think there's some really exciting future opportunities around testing the test deployments of these low-cost soil sensing systems. We also are interested in how we might develop a soil health dashboard that integrates the sensing with the data analytics and the communications. And I think also these soil biology sensors really open up some opportunities to develop exciting new knowledge in a field which has really struggled to kind of get to the dynamics of what's happening within the soil. So I'll leave it there. Thank Ian for the invite, and thank you here for listening, and thank you online for listening as well. And that's just a little picture of some of the team who did manage to meet in person back in September.
John, thank you very much. I'm not sure whether you can hear me offline. We will do a low technology microphone transfer at this point while being socially distanced, and of course COVID negative, if we believe the low cost lateral flow sensing technology that all of us who are in the physical room undertook in preparation for this meeting. I will be monitoring the chat also for questions. So if you're listening online and you have a question, please put your question in the chat. We've got about 10 minutes for questions, and Perhaps if I could start, John, by asking, with the dry down curves that you've been doing the time series data analysis of, whether these have been done with plants in the system or not, and whether having a plant in the system would actually change the dynamics of that dry down, and how and why.
Yes and yes, I think, Ian. So yeah, the sites that we've been using the data from are kind of natural sites, so they have their own vegetation communities there, and so for sure the plants will influence those dry down curves. So I probably should have said dry down curves of soil plant system rather than of the soil itself.
Thank you, John, for the clarification. I did expect as much, but I know that there has been a trend, at least in some laboratories around the world, to actually develop moisture release curves for soil entirely in situ, in situ in the laboratory, with basically taking a repacked soil, putting it into a beaker, if you like, and then baking it in the oven. And obviously, you know, this project is trying to move away from such invasive sampling, moving towards more dynamic, real soils. And I just hope you could comment on that. I can see lots of information coming in the chat, but that might just be the transcript, which is great to see that that's working as well. Technology showing no limits. I'll leave that question with you.
Yeah, I think that was one of the drivers that we had, was to try and get away from sampling. I think the other thing is that I think there are other parameters that we might derive as well, Ian, and I didn't mention, you know, work at the moment we've been looking at is using sensors at different depths, and then looking at trying to get to automate the kind of the travel time of the peak through the soil, so that you might be able to say something about infiltration. Well, not really infiltration rates, because we're not working with water potential. We're working with soil moisture, so we can't get the, you know, the conductivity, but we could get the travel times between different positions in the soil, and that I think would also be another useful parameter that could be derived, you know, from that kind of analysis. We've got a question in the audience.
Perhaps ask the question, Eric, and then John, if you could repeat the question.
Yeah, I think with these sites, you're looking at the soil plant ecosystem, and you're looking hopefully over longer periods of time to detect change. And our kind of first question was could we detect change between very different ecosystems? Do they all look the same? And they don't, fortunately. They do look different. But you're right. I mean, what we wouldn't know is whether it's something to do with the plant, if the plant was, you know, died and the signal changes, or whether, you know, it's somebody who's driven a tractor over the soil and compacted it. We don't know what has happened, and you might need additional information to untangle these signals. But what it would tell you is something has shifted the behavior of that, you know, if you have several years of data, you would expect your soil to be within that data envelope, if it suddenly starts to come out of that envelope or your soil plant system and it starts to appear out of the envelope, you know that there's something shifting. Whether that's shifting positively or negatively, we'll need somebody to interpret the data. I don't know if that answers the. Yeah, that's good. So. Yeah. So the question was around around the soil plant system, and would you like to repeat your question, and then because I'm not going to repeat it correctly. Okay, so the question is that we might have different plants at different locations, which are going to have different water uptake characteristics, and how we might factor that into the monitoring. I think the idea is that the monitoring would run at one site. So you would install your sensors as a station or a monitoring station, and they would continue to supply data, and you would expect that the plant community would be stable. But if that plant community changed, then you may well see a shift in the signal that you're getting back from that site, and that would tell you that something has changed. We're not at the point yet of being able to say exactly what has changed. As I said previously, it could be the soil, or it could be the plant, and it could be the plant community. But we're still. I think the other thing is we're actively trying to work on this at the moment and trying to unravel some of these issues. But it's a good point. Anything online? Okay. Anything else in the room? No, I think we're done.
I think we're done. Thank you very much for that, John. Really enlightening talk. If you could stop sharing your screen, and we've got another speaker to join us. This is Ryan Edge, who's based at Lancaster. Ryan is a wetland ecologist by training, but he's been doing some rather interesting work within a couple of irrigation related projects at Lancaster, and I would invite him to the microphone. And hopefully we can manage a smooth transition from one speaker to the next. So bear with us online while we make this transition.
Okay, good morning, everyone. Anyone online in the room? Am I visible? Can you see the screen? Can you hear me? Perfect. Let me just have a quick look at the technology. Yep, and go back and forward on the slide, so that's good. Good morning. Similar introduction to John there. It's been about 2 years since I've actually stood up in front of people and presented. This is quite a slightly nerve-wracking experience, more nerve-wracking than I thought it was going to be. It's quite good sat at home, but we'll carry on. So today I'm going to be talking about some work I'm doing as part of the SHui project. I've been on the project a few months now, and this is kind of an initial look at the first few experiments that I've been running and some of the data that we got out of that. Talk a little bit about SHui to start off with. It's a really large multinational project, lots of partners across Europe and within China, and it's looking at managing water scarcity. And here at Lancaster University, I, along with a few people in the room, Christina, sat over to my left here, we're really looking at plant soil interface boundaries. And I'm 2 slides in, I'm already saying words like plant-soil interface boundaries, and that's kind of a bit confusing. And what I really mean by that is the bit where the roots and the soil kind of interact, where they touch and come together. And it's quite an interesting kind of really focused area to look at, because it can have quite different properties to the bulk soil and things that perhaps John's sensors are looking at. It can have different kind of chemical properties. It can have different soil oxygenation properties, different water statuses. And that can really impact on plant growth within these systems. You might not pick that up if you're just focusing on the bulk soil. So like I said, it's really important for understanding nutrient acquisition, things like soil stability and erosion as well as plant water relations. So the title of my talk today is, so on the screen at the minute, relationship between rice sheaf development and maize response to deficit irrigation. And I think for many people in the room, they'll know what rice sheaf is, but I think an equal number of people will also be a little bit confused about this term rice sheaf, which is kind of what my research focuses on. So I'm going to take a minute just to kind of go through a few Q&A questions and explain exactly what it is I'm speaking about. So yeah, what is it? It's the soil that's kind of adhered to the root system. So you can see the picture there, and it's kind of the bits of soil that are stuck to the roots. And there's 2 kind of ways that it sticks to the roots, either physically, so it's bound up by kind of root structures, root hairs grabbing hold of it, or chemically by root exudates. And they're the kind of sticky things that the roots exude. And why am I particularly interested in it? And like I said, it's a really promising trait in order to help plants improve plant tolerance to abiotic stress. And specifically drought stress, because it's been shown that the riser sheaf can have a higher soil water content compared to the bulk soil. And you would think that logically, because there's slightly higher water content there, under droughted environments, that would perhaps give a benefit to a plant. And there's been a lot of speculation about this in the literature, and people rightly, or I think rightly, assume that it would, but not too many empirical studies showing this. There's quite a recent one in wheat that's been really good. Came out a couple of years ago now. And we're kind of working on maize. And one of the final questions, probably why maize? And maize has been shown to have quite a lot of variation between varieties in terms of riser sheaf development. And it also fits really nicely with quite a bit of other work going on, different projects within SHui. So we're trying to build this kind of knowledge base and looking at comparable species across projects, I think, or within the project, but across experiments within the project really helps. So a bit about the actual experiment or experiments that I've been running. My research question was really, does increased rice sheaf development improve maize growth under deficit irrigation? And the hypothesis we were looking to test was whether maize varieties with increased rice sheaf development grow more quickly, and did they have a higher tissue water content when subjected to deficit irrigation. To do this, we did a 2-factor experiment. We had a standard or a wild type maize variety and a root hairless mutant. And I used a root hairless mutant, so that's a plant that's been genetically modified so it only grows without, developing root hairs. And that's been shown to lead to a lower riser sheaf development by previous work here at Lancaster University, showing that basically maize plants without root hairs have lower riser sheaf development, and that root hairs are really important for forming riser sheaf physically. I had a 150% of evapotranspiration irrigation treatment, and don't get too hung up on what that means, but essentially 100% is a really well watered treatment, and 50% is a deficit irrigation. So the plant should be quite drought stressed under 50% ET treatment. We repeated this twice. Just for brevity, I don't show all the data pulled together on singularly. It switches between the 2 throughout this presentation. The results are pretty comparable between the first and second run, but the magnitude of the effects is slightly different. So just bear that in mind as I talk through the data in a minute. We had 16 replicates of each treatment, leading to a total of 64 plants. Plants were organized into blocks of 4, and we did 4 destructive measuring time points each week. In terms of the measurements that we took, so the things that we were trying to look at within the plants, we measured evapotranspiration, and that was to kind of monitor the drought stress that we were imposing on the plants and maintaining our watering regimes. That was measured just by simply weighing the amount of water lost from the pots each day using a balance. We also took stomatical conductance readings using a barometer. Really good. We did some then destructive measurements. These were done once a week where we took 16 plants. We measured leaf water potential using psychrometers, and we take a little disc, we put them in these machines, and we measure basically how much water is in the leaf. We do leaf ABA, which is a hormone. Not the best picture of leaf ABA analysis there, but you sample a leaf and you go away and measure it. And then we also measured the riser sheath as well. And to top it off, we wanted to get some general measurements, kind of plant growth, things that are more familiar. There was a lot of chopping up plants, putting them in paper bags and drying them for above ground biomass and root mass. In terms of results now, this is kind of a sanity check. And what we're really interested in here is that the blue and the green lines are higher than our yellow and our orange lines. And from that, we can see that the blue and green lines were more well watered, higher water status in the pots compared to our yellow and orange lines. So we did have a deficit irrigation or well water treatment. And our other thing is, I told you before that root hairless plants should have a lower riser sheath development, and kind of a sanity check there. Well, yes, our plants with root hairs had more riser sheath than our plants without. So we had our well watered and our deficit irrigation treatment and our low riser sheath, high riser sheath treatment as well. In terms of some kind of plant physiology, our above ground, we didn't find very many differences between treatments. The only thing that we could see is that plants were getting bigger over time and our well water treatment was bigger, but no difference between our genotypes, and that would have been our high riser sheath, low riser sheath variety. So we didn't see much discrepancy between them in terms of above ground biomass. We did see a difference in terms of root hairs. In terms of leaf area, our root head variety, so our high riser sheath variety, had higher leaf area than our low riser sheath variety, and our well water treatment more than our deficit irrigation treatment. But I think the key here is we don't really discriminate under kind of drought conditions. So the two red boxes on the right hand side there are deficit irrigation treatment and our high riser sheath, low riser sheath variety. And in the pairwise comparisons of our stats, we really aren't able to pull apart any difference there. So it's quite difficult to say that, yes, having a high riser sheath is helping you grow bigger under a deficit irrigation treatment, at least in terms of leaf area, which is quite a good indication of growth, I think. One other thing that we measured was kind of leaf water potential I spoke about earlier. And again, we weren't able to reliably differentiate between our treatments. And all that we see is that all we can really take from it is it decreases over time. And we would expect that as the plants get slightly bigger, the rate of evapotranspiration increases, and therefore the plants are slightly more drought stressed as we're measuring them. But yeah, no difference between our treatments. Leaf ABA, which is a hormone particularly related to drought stress, again, we didn't differentiate between our treatments, but it increased over time, as we would expect with leaf water potential decreasing and rate of evapotranspiration also increasing as well. Conductance, again, really similar story, so decreasing over time, and these are all really interlinked. We would expect as leaf ABA goes up, stomata conductance. And the stomata to close as plants are getting more drought stressed, but again, we're not differentiating between our treatments. And overall, that's not the most super interesting set of results, given the really interesting hypothesis I think I posed at the beginning of the experiment. I think it's important to acknowledge straight off the bat there was an awful lot of variation. There's come quite wide bars and box and whisker plots that I'm showing you there. Quite a lot of anomalous points within the graphing software I've used. And that's making it quite hard to differentiate between the treatments and the genotypes, I think. And it's something that's going to have to be assessed as we move forward in the project. There's some very simple conclusions we can say. Well, water plants tended to grow a bit bigger, so no surprise there. And root hairs tended to grow larger as well. But I think the key there is that the root hair varieties weren't performing significantly better under drought stress as they were well watered. And root hairs are important for a lot more than just riser sheath development and drought stress, there are also important things like nutrient acquisition as well. We did see that our root hairless gene types had less riser sheath. So when we come back to kind of my original hypothesis, we definitely reject that. I mean, the data we have really doesn't substantiate what we were thinking. There's a really important part of this that I haven't spoken about yet, and the reason I chose to kind of talk to you guys about this today rather than some other research that might be a bit further along that I've done previously, and that's the term development. So I did multiple weekly time points, and I was really interested in how rice sheath develops over time. So a lot of literature that you look at in terms of rice sheath development, it tends to be like single time points, and you don't get that kind of how is this growing in plants, how is this thing, it's a dynamic trait, or is it a dynamic trait? We don't know. So this is the same data I showed earlier when I just showed yes and no, but split between the treatments and over weeks. And overall, you see, it's quite stable. If you try and follow a block round, so taking the first pot, for instance, it's the first box on each graph, it's slightly bigger in week 2, and then weeks 3 and 4, it's very similar. Again, with the second plot and second box going along each one, it doesn't seem to change very much other than like a peak in week 2. And just to help you guys visualize that, if I plot the same data, but I just combine all 4 of those little bars into each week, you can see, yeah, it peaks in week 2, but it's quite stable over time. And if you think about it, that's slightly odd, because you would expect, you know, things like above ground biomass got larger, root mass, which I'll show you in a second, gets larger over time, leaf area increases over time. You would expect, you know, as plants grow bigger, things would increase, and our riser sheath really isn't showing that, particularly if you look at this in relation to root mass. So what we're seeing here is actually that as root mass increases, the amount of riser sheath per amount of root decreases, and that's quite odd. So you would expect more root to have more riser sheath. There's more area for soil to stick to. You'd expect to have higher riser sheath, and that's usually how it's plotted when you see it in papers and stuff like that, is that you see riser sheath divided by root mass is what people usually look at, and that's because it's kind of a normalized value and allows you to look at differences between treatments at different sizes and things like that. So what you see in ours is that actually riser sheath is decreasing, riser sheath divided by root mass is decreasing over time, and this also appears to be higher in our root hair varieties, which are the green lines, compared to the blue lines. So our root hair plants with root hairs that start off with their larger plants, larger amount of riser sheath, actually lose a great proportion of their riser sheath per root mass as the plants develop. And this could also explain why we're not seeing these expected benefits of riser sheath on plants' drought tolerance, and that's because the drought stress is slightly increasing over time as the evapotranspiration increases over time, but the actual amount of riser sheath relative to the plant, which is the thing we were looking at providing a benefit, decreases over time. The study really wasn't set up initially to look at this, so these are limited kind of things we can take in terms of conclusions. So we can say that riser sheath relative root mass decreased over time, and I think that's really interesting in of itself. And the drop was steepest in the larger plants with root hairs, but those are generally bigger plants as well. And I'm not the world's best statistician, but as far as I can tell, it's difficult to say from my data whether that is a function of just having larger plants, so it's easier to discriminate the steepness of that slope compared to the smaller plants without the root hair. So I think there's some more work to be done on that as well. And also whether root hairs in themselves are the main drivers of this. We know root hairs in a lot of other species are quite a transient trait, so they're not permanent on the roots of the plant. They change off. The amount of density of the root hairs is different on different parts of the root system. And we know that plants without root hairs actually develop slightly different root systems to plants with, and almost as kind of a compensatory mechanism. So there's some really interesting work to be done moving forward with this. And yeah, that's as far as I've got with it currently, and I'm looking forward to doing more of it in the new year. Any questions?
So if we've got some questions for Ryan online, please put them in the chat. Otherwise, if you're in the room here, please raise your hand if you have any questions. On.
Yeah. Repeat your question and then I'll. Yeah. So John asked whether the declining relationship between riser sheath over time is that riser sheath develops really, really quickly and then is a relatively static trait. And the answer to that is I really don't know. And it's something I think needs to be looked at in a bit more detail. The problem I have is the size of the plants. Right at the beginning, they're quite variable within each other. So trying to normalize for that over time is hard. One thing we do know is quite important is that you need wet and dry cycles in order to form riser sheath. I'm not entirely well up on the physics of that. I know there's people in the room like Billy's nodding away here. He might be able to tell you more about that. But the wet and dry cycles are super important. So I'd imagine they need to have at least a few wet and dry cycles early doors. It's going to take maybe more than a week where we were seeing that increase in the drop. So something to look into.
So returning to questions that have been posed online, we have a question about whether the exudations or the root exudates from the different genotypes differ and whether that's something that you've measured and perhaps you could speculate what might be important to measure here.
Okay, I haven't measured it. I know, was it B? B is a previous PhD student at our university has. And without having a paper in front of me, I don't want to go say exactly the wrong thing, but I can definitely post that in the chat in a minute. I can find the paper. One thing I know that B did find, and I think a key result from that is that root hairs were a lot more important for riser sheath development than root exudates. They had a much greater proportional effect. So that's why we were looking more at root hairs than root exudates in this study, or that study as well.
We have a methodological question that has been posed online, and that concerns how you actually measured the riser sheath, given that this could be quite technically challenging, and wondering whether there is any mechanical disturbance needed to actually capture the measurement of the riser sheath.
Okay, I think anyone that's measured riser sheath knows it's completely imperfect method, and we actually have a working group at Lancaster University at the moment trying to find a better solution for doing this. But essentially you have to physically remove the plant from the pot, shake off the excess soil, and then you're left with what's stuck to the roots. And how you measure that differs a little bit between members of our lab group, but essentially you're washing it off the soil and measuring either the soil that you've washed off or the roots, that the difference in weight between the root that you had before, the dirty root, and then the clean root that you had after. But like I said, if anyone has different ways of measuring riser sheath or interested, please email me. You can join our working group because we're trying to solve this problem.
So a relatively straightforward question now. A lot of interest in the growing medium that you used, and also perhaps wondering whether the extent of riser sheath development is determined by the soil texture.
We used topsoil from Norfolk in the UK. We use that because quite a lot of members of our lab group do it. People that worked on riser sheath within our lab group have used it before to kind of make sure our results are comparable between each when we're looking at it. We know for a fact that different soil textures have a really big impact on riser sheath development. There's been a few papers out recently looking at that. Ian, do you know? There was one you sent around the other day. It was good. Either way, we know it has a big impact on riser sheath development, and I think that's a really active area of research going forward that we're probably going to look into as well.
And a final question from the chat. simply whether the proportion of the roots that have root hairs, whether that determines rhizosheath development. I mean, my understanding is that all root classes of the maize plant will develop root hairs, but the proportion of the different roots that each plant has may have an impact on rhizosheath development.
Yep, and I think that's probably where we're going to go moving forward with this project, is really looking at differences in root structure, including root hairs, lateral roots and things like that, and how that develops over time. Because I think that's another key point that we can't pull out of our data, which is probably quite interesting. A plant's root structure at 1 week old, in terms of the proportion of different roots, is probably going to be quite different to a plant's root structure at 4 weeks old, and whether that can explain, or then 8 weeks, 12 weeks, that kind of thing, whether that can explain some of the differences that we're seeing within our study would be very interesting.
Great. Thank you very much, Ryan, for your presentation. I'll just let you stop sharing your screen and go for the changeover for the next speaker. We'll have to take any additional questions at the coffee break, which is coming up in about 40 minutes time. So thank you very much for that, Ryan. So we've had a nice measurement about how we might measure the soil, and we've had another nice presentation about how we might measure soil that is at the interface with the plant. And clearly, I think there's a lot of scope for some methodology development here, given some of the challenges we've heard of measuring both soil and soil plant interactions already this morning. So we'll move now to our next speaker, Billy, who is a researcher here also at Lancaster. Billy's going to talk to us a little bit about his PhD research, which is quite an applied project looking at the impact of surfactants on plant-water relations. So Billy, thank you very much. I'll pass over the microphone to you.
Hello everyone, can you see my screen? Can you hear me? Great. So as Ian said, today I'm going to talk a little bit about some of the work that I have done during my PhD, where I was investigating the effects of soil surfactants on plant-water relations. So I'll start with a very brief background about how we ended up with this research and how it fits in the bigger picture, and also with the theme of this symposium. So as we already know, due to climate change, water scarcity is a global threat. So there are studies that suggest that by 2040, large parts of the planet will live in areas where drought stress will be the norm and not the exception. And also this is a challenge also for countries like the UK, where it usually rains a lot. Today it doesn't, but anyway. However, for instance, this summer of 2021 was quite dry for the largest part of the country, as you may see also in the map on the right, similarly to summers of 2020 and 2019 at least. So there are also other studies, well, a lot of them actually, which suggest that in the next couple of decades, parts of the country could run dry as well. So it's vital to manage our current agricultural systems in a global level. Actually, in order to ensure their resilience, especially since the population of the planet is rising. So application of surfactants to soil may be part of this solution, let's say. However, further research is needed to investigate whether they could be used more broadly in arable crops to enhance resource use efficiency, such as water or even nutrients. So as surfactants, we define amphiphilic molecules that decrease the surface tension of water. They are amphiphilic because they possess both a hydrophobic and a hydrophilic part in the same molecule. So essentially, hydrophobic tail hates water and hydrophilic head loves water. In agriculture, they have been used mainly in the tough gas industry as wetting agents to mitigate against soil water repellency by increasing infiltration rate. And also in some other cases, it has been found that surfactants can also enhance soil moisture distribution within the soil profile. However, during the last few years, there is an increasing interest in using surfactants to other arable crops to enhance acquisition of water and nutrients. There are some interesting studies. For instance, in one of them, in maize, it has been found that applying surfactants to soil increased total dry matter and grain yield under 4 different irrigation regimes. So when plants were watered, as well as when they were stressed. However, the effects of surfactants on plant physiology remain unknown, and thus the aim of this research was to investigate if and how surfactants can alter soil and plant water relations and understand whether they might be used as a tool to mitigate against water scarcity. So to do that, the first thing that we wanted to understand better is whether surfactants can affect soil water status. To do that, I constructed soil moisture release curves using psychrometers, as you may see in those 2 pictures of a sandy soil that I was using for my experiments. And as soil moisture release curves, I mean the relationship between soil moisture and soil water potential. What we've actually found is that for the same soil water content, surfactants increased soil water availability. So basically, this means that as soil was drying, surfactants made soil water more available to plants, which was the first positive effect that we have seen. So these differences in soil water availability may be attributed to decreased matrix forces due to surfactants action in the surface tension of water. Decreased matrix forces would make water to be held less firmly in the soil particles, and that would make soil water more available to plants. To investigate this further and actually confirm our this speculation, this assumption, we determined the effects of surfactants on matrix forces indirectly, not in situ. So to do that, we monitored soil moisture recovery following basal rewatering in 2 soil layers, upper and lower, using soil moisture sensors in 2 species, in barley and maize plants that were growing either in soil with surfactant or without as a control. And the main outcome from this study was that in both species, both in maize and in barley, surfactants hindered rehydration of the upper soil layer, as you may see, which was also a significant response. So these results may indicate that actually surfactants had an effect on matrix forces, and actually they decreased matrix forces, which may explain how they made soil water more available to plants. Or at least this could be one possible explanation. A second possible explanation about these differences in water availability could be rhizosheath formation. So as Ryan mentioned before, as rhizosheath, we define soil particles that are adhered to the root surface due to the action of mucilage and root hairs, of course. So what I've done is that I measured rhizosheath formation in barley plants that were growing either in soil with surfactant or without, again, as the control. To do that, I followed the typical method that is described in the literature. So I excavated the roots from the pots, I shaked them gently, and I collected adhered soil particles in trays full of water. I oven-dried those trays, and the dry weight of the soil is defined as the rhizosheath weight. To normalize those data, I also measured root length using a scanner, a root scanner. So the data that I'm presenting today in this graph are specific rhizosheath weight, which means rhizosheath per unit of root length. And the main message here is that surfactants actually amplified rhizosheath formation in a barley, which has also been reported previously. And the possible explanation, according to literature, is that surfactant molecules actually interact with the mucilage from the roots, and due to this interaction, a cross-linking is occurred. Therefore, mucilage is more viscous, and it remains in the vicinity of the roots in higher amounts, and therefore more soil particles are adhered. This is what has been described previously, as I've said. However, I haven't tested that. Furthermore, based on these results, we could say that surfactant-induced rhizosheath formation may also facilitate root access to water by retaining a better contact between soil particles and the root system, which could also make soil water more available to plants. So this also might be a second explanation on how surfactants made soil water more available. The other question that we wanted to answer is whether these positive effects of surfactants on water availability of the soil could have any impact on the plant, on leaf water status specifically. To do that, I used again barley plants, since these were my main model species. And I measured leaf water potential using a pressure chamber, as you may see in the picture on the right, to quantify actually how stressed the plants were. I did that during a drying cycle, so when the plants were well watered in graph A, as well as when they were stressed in graphs B and C. As you may see, when plants were well watered, which is the graph A, there were no differences in leaf water potential between the two treatments. However, when plants were growing in mildly dry soil, which is figure B, we've seen that surfactants increased leaf water potential. This basically means that plants that were growing in soil with surfactant were less stressed than the control ones. However, those differences were eliminated when plants were growing in severely dry soil, which is graph C. And yeah, the next slide, it's actually the last data set that I'm presenting. So it's some nice, some interesting actually results in case of plant growth in shoot fresh weight and shoot dry weight. So in one of our last experiments in barley and in maize, we found that surfactants significantly increased shoot dry weight for both species, as you may see over there, and also tended to increase shoot fresh weight, even if this second one wasn't significant statistically, which was quite interesting and promising. However, at least until now, we are unable to explain why this happened, because we couldn't actually link those biomass differences with physiological measurements that we conducted, measurements of photosynthesis and leaf water potential, at least during the times when we conducted those measurements. And therefore, further work should focus on understanding mechanistically how surfactants increased shoot dry biomass in those two species. So to wrap up with some main conclusions, we may say that probably surfactants may enhance ability of plants to acquire water, since we've seen that they made soil water more available to plants. This may be attributed either to decreased matrix forces or to amplified rhizosis formation. Either way, we've seen that due to these positive effects of surfactants on water availability of the soil, they also enhanced leaf water status with moderate soil drying. So those plants were less stressed than the control ones. And finally, in one of our last studies, we've seen that surfactants significantly increased should dry biomass. However, we couldn't explain mechanistically why this happened. And in terms of future work, I think that more field trials should be established to understand further which is the agronomical significance of surfactants on biomass as well as on yield. Yeah, so that was me. Thank you for listening. And if you have any questions, I'm happy to answer them.
Thank you, Billy. As before, if you've got questions online, please place them in the chat. Perhaps if I could just start with a question, Billy. It was quite interesting that both the maize and the barley showed increased biomass with the addition of the surfactant. Yet my understanding is that the exudates released from the root system are very different between barley and maize. I think there's been some nice work from Paul Hallett's group in Aberdeen showing that the barley plants release exudates that are high in organic acids that are quite effective in disaggregating the soil, whereas the maize plants release a much more polysaccharide-rich exudate that's actually quite good at aggregating the soil. And so I'm interested to know whether you saw differences in rhizosheath formation with the addition of surfactants in these two different species.
Thank you, Ian. Basically, I measured rhizosheath formation only in barley, not in maize. So in case of barley, as I've said, surfactants increased rhizosheath formation. This was a positive result. However, I didn't measure rhizosheath formation in maize, so I don't know the answer. Probably surfactants may have behaved differently because they, as you said, maize has different molecular properties. So this cross-linking may not be occurred, but this is something that should be studied.
So we also have a question online about how feasible it is to conduct experiments in the field with surfactants, and some interest in knowing how these surfactants might actually be applied. I guess it's reasonable to do trials of the surfactants, but I wonder whether a cost-benefit analysis would actually show that they could be important.
So surfactants actually have been applied in the field in case of turf grass field trials, where it's actually the main way that surfactants are used currently in turf grass courses, in general in turf grass fields. So the way that you apply them actually, I think, is using big bottle sprays. So you apply them to the soil before the plants have been established, and at least in turf grass, I think that you need to reapply them every 3 or 4 months, because during time their action is reduces. And there was one more thing about economical evaluation, I think. Yeah, actually there is one study, which is a few years ago in case of maize, I think, where they've conducted an economotechnical study. And I think that they've seen that applying surfactants, of course, is an additional cost, but the additional yield that you will have could be even a greater benefit. So it might be beneficial to use them.
Got one question from the audience, Cynthia. So the question was, at what stage of development did you measure, what stage of plant development did you measure the rhizome sheath in these plants? And did you also see the rhizome sheath development stabilizing over time in your experiments? So we'll let you address that question, and then any other questions we'll have to defer till the coffee break, because we've got one more speaker coming up this morning before the coffee break.
So I measured rhizome formation in the barley plants that were roughly 3 weeks old. So I didn't measure them within the development stages like Ryan did. So I don't know if before and after rhizome sheath would decline. That's something that should be addressed. And also you had one more question about biomass in maize and barley. For my last slide. So those plants were roughly 4 weeks old, both for maize and barley.
Great, thank you very much, Billy. We'll let you unshare your screen in preparation for the next speaker. And now we are going to try something completely different and trust the technology. And we're going to have a speaker online. So Katharina Huntenburg, who is currently at the National Institute of Agricultural Botany, is going to be our next speaker. And she's going to talk to us about some interesting work imaging potato tubers. I think I can see you on the screen, Katharina. Are you able to share your screen?
I'm trying. It says loading your presentation. So I hope we will be there soon. Yes. Can you see the screen?
I can see the screen now. And so whenever you're ready, Katharina.
Lovely. Thank you. Well, thanks for inviting me, and thanks for the speakers before me, because they've covered a bit of what I couldn't say in the next 15 minutes. So that's great. For example, why do we need to think about drought stress in a country like the UK that is known for a lot of rain? Anyway, I am working on potatoes. And my topic is using an MRI to quantify the water content of the potato tubers and of the in drying soil. And how did I come to that? I did a field experiment in 2018 with soil compaction and drought stress. So yeah, I tried to change the soil conditions and see what's happening to the plant. And you can see on the right-hand side, can you see my cursor? No, cursor, let's put that, maybe that. Anyway, on the right-hand side, that's the control plant, uncompacted, well-watered, and then you can see all the other combinations of stresses are smaller, and I've also had a lower yield from the other stresses. So lower water availability leads to smaller plants and lower yield. So far, so predictable. The question is why? And I looked into gas exchange and water relations. Similar to Billy, I did not see differences. So there's no difference between the treatments in assimilation rate, which means also that transpiration rates were similar, which is a bit surprising, I thought, because they didn't have as much water, some of them. So I thought, okay, if they are in drying soil and transpire similarly, they should have a different leaf water potential. But no, that was the same as well. So that led me to ask, how does a potato plant cope with drought stress or with a reduced water availability? And in the controlled environment, we did indeed see that they do close the stomata when they experience drought stress. So maybe there's a difference between the long-term and the short-term reaction. But I also thought, well, we've got this huge storage organ of the tubers, and maybe they have an impact on the total water content of the plant as well, and on the water household of the plant. So we started looking into that a bit. And firstly, I looked into the literature, and it's actually really hard to find something on the tubers because it's really hard to measure something that's underground. But there is a study from the 70s that shows these lines are water potential of the leaf and of the tuber in the field. So they dug out tubers in the field and measured leaves of the same plant. And there's two things that we need to note here. So one thing is we've got a wave pattern. So every gray bar is a nighttime measurement. So there's a wave pattern. Water potential drops in the leaves and the tubers in the daytime and increases again, kind of recovers again overnight. For the leaves, that's what we expect. It's I wouldn't expect it for the tubers because so far, or from what I've learned, the tubers are always just a sink organ, so everything goes into the tuber, but nothing goes out of it. And this indicates that there is something happening with the water content in the tubers. And the second thing that we can see is that the leaves on the tubers, that the water potential of the leaves is always lower, so more negative than the one of the tubers. That means that there is a chance that water is flowing from the tubers to the leaves. So far, so good, but this is these are single tubers that have to be dug out, and afterwards you can't measure them again because they are detached from the plant. So we wanted to look into that a bit closer, and there's a great technology, MRI, that can image tubers in the, or organs in the soil. And the Forschungszentrum in Jülich in Germany, they do have a plant MRI. Looks like, well, that's the robot that brings them into the MRI later on the right hand side there. And see if it, yes. So the MRI images protons, and in plants, that's mainly water molecules. That's why from the image we can deduct one thing that we can see is the shape of the tuber, as you can see in the image up there, and deduct the tuber volume from it. And then we can see the signal strength, and that is the tuber water content. So we took measurements every 4 hours of our plants, and we had well-watered ones and drought-stressed ones. The well-watered ones are the blue ones, and the blue line, and the drought-stressed ones are the orange line. And in both, what we can see is, again, this wave pattern that we saw in the previous publication on water potential. What we can also see is that the water content in general in the well-watered ones steadily increases, so over time. So what's happening is overnight the plants recover or recharge the water in the tubers. In the daytime, it does look a bit as though it's decreasing. It's not statistically significant, unfortunately. But then they recharge again overnight or fill up with water to a higher degree than the night before. And then this whole game starts again in well-watered plants. In the drought-stressed plants, we still have a bit of a wave pattern, but if we start at a value of 1 in the beginning, then we go down a bit during the day in the water content, and at night the water content recovers, but it only recovers to about the same value that we had when we just started. So it's not higher than that. Then in the daytime, it decreases, and this is a significant decrease. And then it recovers again a little bit overnight, but doesn't reach the same values as the night before. And then only when we rewater the plants, we have an increase again. So what is, oh, let's see. Can you see this? Well, anyway.
Yes, that's fine.
Oh, that's good. Then I'll try this. Just seeing this on the side. Right. So what is important to note here is, or why is it important that the value in the drought-stressed ones, even though they do the water content does increase overnight, it only reaches the value 1 that we started with, is tuber growth happens either through cell division or through cell elongation. And cell elongation can only happen if the turgor pressure is high enough in the cell to expand the cell. So if we lose some water out of the cell, all the cells in the daytime, and then there's some water coming back in that kind of brings them back to the turgor that they had, but it's not going over that. So we go back to where we started, but we're not going higher than that. So the turgor is not high enough to make the cells expand. Whereas in the well-watered plants, it does go well above the values that we started off with. So there is enough pressure in those cells to do the expansion and to actually lead to volume growth of the whole organ. And that's exactly what we're going to see in the next slide. Oh, that's daytime where the stomata are open, and nighttime, forgot that I had that. Okay, here we go. So this is tuber volume. And what we can see again is a wave pattern in the well-watered plants. It's not quite as big, the waves are not quite as big as in the water content, but generally what is happening is we have an increase of volume overnight, and that's exactly what I said earlier. The water content in the cells is increasing, and hence the cells do expand because the turgor pressure in the cell is higher than it was before. Then it looks almost as though there's a decrease in volume in the daytime, but again, this is not statistically significant. And but we have an increase in volume again over the night. In the drought-stressed plants, it almost looks as though there is a little bit of an increase in the first night in volume, but this is not significant. And again, all these like wobbles in those 2 days where the plants do not get water are not significantly different from each other, which means we have zero volume growth as soon as we stop watering the plants. So this is a very mild drought stress, but so yeah, and maybe there is a little bit, maybe with more samples, there would be a little bit of an increase in volume, but in general, they just stop doing anything. And that is exactly because the water content of the tubers or of the cells is not big enough for that volume growth, for the cell expansion. And as soon as we rewater them, the volume goes back up. It doesn't quite reach the levels of the well-watered ones, which also poses the question a bit, for how long can we stress the plant, and can we not water them to make sure that the growth of the organ does recover? Because what we are interested in the end is tuber growth and yield. So that brings me to the summary again already. So what I found was tubers do lose water over the day, and there is water going from in the daytime. We have open stomata. There's water from the soil going through the whole plant, through the stomata, and back out in the atmosphere. But there seems to be also some water leaving the tuber, and it's highly likely that this water is also going into the transpiration stream and leaving the plant that way. Overnight, the water content is recharged, and this recharging is much stronger in the well-watered plants just because there's more water available in the soil than in drought-stressed plants. And that happens because at night the stomata are closed, so the leaves do not need as much water. They just kind of recharge their resources, and after that, all the water that is available and that the plant can take up at night is directed towards the tubers. And that means that tuber volume growth happens overnight. So in the well-watered plants, tubers grow overnight, stay similar over the day, grow again overnight, and that's how we get to bigger tubers. But it stops immediately in drying soil. So nothing happening as soon as we dry the soil out, and then a recovery as soon as we rewater. And again, the implications for farming might be that we need to think about how close we need irrigation intervals. Would also be interesting to look at different potato varieties, because I've always worked with Maris Piper here. So all that leaves me to say is thank you, Ian, for supporting me in pursuing this slightly off-track project during my PhD. Thanks to the colleagues in Jülich who facilitated my work there, were really helpful. I got EPPN 2020 funding for running the experiments over there, and I can only recommend people who want to do phenotyping, check this network out. It's amazing what kind of facilities are out there, and there is funding to go there and use them. Was a great experience. Yeah, can only recommend it. And thanks for AHDB for funding my PhD. And yeah, I'm happy to take questions.
Hopefully, Katharina, thank you for that. You saw some virtual applause on the screen, but also might have even heard some actual applause from the physical room here. So do we have any questions for Katharina, either virtually or physically? So we've got a question in the room here, Katharina. Ryan, do you want to say that, and I'll repeat it to you, Katharina? So I think I've understood the question. Please nod if that's the case, Ryan. So the growth of the potato tuber overnight, how much is attributable to water uptake and how much is attributable to the division of new cells?
I think in this case, it's mainly attributable to the water uptake. I did not measure a difference in dry matter in the tubers, and the time frame is relatively short. I mean, it was only for 3 days. Yeah, but I think it would need more samples to be really sure about that, and maybe a longer time period to check that.
Perhaps I could ask a follow-up question, Katharina. How, I mean, in terms of the size of a potato tuber, how long do the cells divide for? Does that continue up until full growth of the plant, or is there a defined period of cell division and then later a defined period of cell expansion?
That's a really good question. I don't know, is the short answer. I would imagine that both is happening at the same time, because the tubers that we were measuring in this experiment were relatively small. So that was like a week or two after the initiation. So after we could see the first like little bubbles at the end of the stolon. So they were like relatively small, I would say, about, if you can see it, about that size. And since we can see an effect of cell elongation at that time point, and they will grow to like proper tubers of that size, I'm relatively certain there must be some more cell division going on as well later on in the development.
I think it's a really interesting system that you've got to work with, and particularly challenging. And given that you're based at an institute that is focused to a large extent on potato, and you've got plenty of time to address some of these questions, I think we'll take one more question from within the room here. John. I'll just answer quickly. Yes, potentially, John. And this is something that Katharina wrote about in her PhD thesis. So, Katharina, the question that John's addressed is whether you're better off irrigating the potato plants at night to potentially maximize the tuber development and their growth. So I'll let you answer in your own words, having perhaps given a rather presumptuous answer.
Yes, it's something that I suggest might be worthwhile. And we are actually thinking about setting up a project with drip irrigation in the field with potatoes and letting the drip irrigation run overnight. The problem with nighttime irrigation, as it is done at the moment with spray booms and guns in the field, is that you get a long leaf wetness period, and then you get difficulties with blight, which is a fungal disease. And if the leaves stay wet overnight, blight is more likely to come in. So it's not really feasible, but with drip irrigation, we wouldn't have that problem. So we are thinking about instigating a project. So if anyone is wanting to join that sort of project, please drop me an email. I'd be interested to hear what you have to say or what you want to do. It's something that we're looking into. I'll put my email address in the chat. I forgot to put it on the last slide, so that you know how to contact me, or just contact Ian and he'll just forward it.
I'll certainly be sending you an email, Katharina. It's fascinating to show how, you know, a very fundamental research question that you've addressed using a very novel technique might actually have some practical application if irrigation can be applied at the right time and in the right form to a very commercially relevant crop. So thank you very much for sharing that with us. Unfortunately, though, you won't be able to share the coffee break with us, which is where us in the physical room will be going to. So this brings us to a close of the first part of this symposium. We will be reconvening in 25 minutes time. So if you've been sitting at home listening to this or in a research institute, please take the time to stretch your legs, get some exercise, perhaps caffeinate yourself, and we'll be reconvening in 25 minutes time. So thank you very much for your attendance, and a big thank you particularly to all our presenters this morning. I've found it an incredibly interesting morning. So thank you very much for now, and we will see you shortly in 25 minutes time. Okay, bye for now.
How do I stop sharing this?